Pollen preparation is a well-established method. Most universities and laboratories follow the Moore et al. 1991 method with some minor modifications depending on the type of sediment used. Stage 1 is sampling. Before you begin to sample, you will need a tape measure, a scalpel, a volumetric sampler, a beaker or a bag to keep the samples in, and a marker pen. Before you begin your sampling, it's also a good idea to make sure that you have described and photographed your core. This will help you interpret your results. The resolution at which your sediment should be sampled should be discussed with your supervisors. Once you have decided where you are going to take your samples, first you need to label the beaker or bag that you're going to put your sample in. Write as much detail as possible, the core name, sample number and the depth. In order for your results to be comparable, you must take 1 centimetre cubed of sediment. The way that you do this is using a volumetric sampler. This is the standard for pollen analysis. To do this, take the tube part 2 and place it onto the base part 3. Place the entire sampler into the core, twisting the sampler carefully as you do so. This will take the majority of your 1 centimetre sample that you will need. Then you need to remove the sampler from your core and using a spatula fill in any of the remaining space in the chamber. You want the chamber to be completely full to make sure you have a whole 1 centimetre cubed. Use the final part, part 1, to just push down the top of the material to make sure the chamber is completely full. Finally, using the part 1 once again, pop the entire sample into the beaker by pushing it through the tube. Rinse all your utensils thoroughly in between each sample. The next step is to add hydrochloric acid at 10% concentration. Ensure you are wearing the correct PPE before you begin and carry out this step in a fume cupboard. To be able to determine the concentration of pollen in the sediment, we add an exotic pollen tablet with a known number of exotic pollen grains. In this case, we are using Lycopodium. Ensure you note the batch number when adding the tablet, as each batch will have a slightly different number of grains per tablet. Add 1 tablet to each sample. The HCl will dissolve the Lycopodium tablet and also digest any calcium carbonate in your samples. Add it very slowly because it will react and effervescence will occur. Keep adding HCl until the reaction has completed, then fill the beaker with deionized water. Your sample is now ready for sieving. The sieving stage removes anything that is too big to be a pollen grain or anything that is too small to identify. For the largest sieve size, we use a 125 micron sieve, and for the smaller, we use a 10 micron sieve mesh and frame. This is the equipment that you will need. Begin by labelling your tubes with all the information that was on your beaker. You then need to rinse all of your equipment in deionized water before you assemble it. Rinse the mesh and then place it into the sieve frame. Then rinse your 125 micron sieve and place it on the top. Make sure you hold the 2 parts of the sieve together securely to stop the bottom part falling off into the sink. Pour the entire sample into the upper sieve using deionized water. Add deionized water while agitating the samples with a plastic stirrer. Continue to wash the coarse fraction until all fine particulate matter is suspended in the lower sieve. Check that the lower sieve doesn't overflow. Remove the coarse sieve and rinse with deionized water. Then, using the rounded end of the sieve stick, continue to agitate the material, washing through the fine mesh to remove the fine silts and clays until the water runs through clear. When you are finished, rinse all the material to the bottom of the sieve frame and, using a funnel, pour it into your labelled tubes. Disassemble and rinse all parts of the sieve in deionized water. Once they are clean, you can assemble everything again ready for the next sample. Mesh can be used up to 4 times, providing that it is cleaned properly between samples to avoid any cross-contamination. At Royal Holloway, we use the flotation method using a heavy liquid called sodium polytungstate, or SPT, to remove the mineral fraction from the pollen sample. This step is necessary to get the pollen slides as clean as possible, enabling accurate analysis. If the sample is high in fine minerals, we substitute this step for the hydrofluoric acid treatment. First, you need to centrifuge your samples for 5 minutes at 2,500 rpm to concentrate the sample at the bottom of the tube so we can pour off the water. Next, you need to make the SPT to the density you require. For pollen, we use 2.0 grams per centimetre cubed. Using a hydrometer, check the density of the liquid. Add deionized water and mix carefully using a stirrer. You then need to check the density again. Repeat this process until the desired density is achieved. Read the scale of the hydrometer at the bottom of the meniscus of the liquid. Add 5 ml of sodium polytungstate to each centrifuge tube and agitate using a whirly mixer until thoroughly mixed and disaggregated. Set the centrifuge for 20 minutes at 2,500 rpm. During the time the samples are spinning, you can label some centrifuge tubes, ready to decant the floated sample. When the samples are done in the centrifuge, take them out very carefully so you don't mix the organic and inorganic sediment. You will easily be able to see the 2 different levels of material. Pour off the organic suspension from the sample into your new labelled centrifuge tube. Top up the sample with deionized water and whirly mix once again. Place the samples back into the centrifuge for 5 minutes at 2,500 rpm. Once they are finished, pour off the supernant into a beaker. The SPT can be recovered using evaporation and filtration. Wash your samples at least 5 times using a cycle of deionized water and centrifuge to make sure there is no SPT left before you move on to the next stage. This stage of the procedure involves highly corrosive chemicals and must be done in a fume cupboard with the appropriate PPE. A red warning light outside the lab must be switched on to indicate that acetylosis is taking place and other lab users should not enter the lab during this stage. The acetylosis removes soluble cellulose in the samples whilst preserving the pollen, spores and charcoal. The first step is to dehydrate the sample. This is a very important step because if there is any water left in the sample at all, it will explode in the next stage. Fill each tube with approximately 10 ml of glacial acetic acid and whirly mix to make sure the whole sample is covered. Put the samples in the centrifuge and spin for 5 minutes. Pour off the supernatant slowly into running water in the sink located inside the fume cupboard. Leave the tap running. Next, you need to make up the acetylosis mix. Each sample will need 5 ml of mixture. Calculate the volume needed of each chemical using the ratio of 9 to 1. 9 parts acetic anhydride to 1 part of sulfuric acid. First pour the acetic anhydride into the conical flask, then slowly add the sulfuric acid whilst swirling the mixture. It's important to do this slowly to stop the mixture burning. Be aware that there is heat produced during this process. Add 5 ml of mixture to each sample and whirly mix. Place the samples in a boiling water bath for exactly 3 minutes and 30 seconds. Any longer than this, the pollen and spore grains will be destroyed. Take the samples out of the bath and centrifuge for 5 minutes. Once centrifuged, pour off the tubes very slowly and carefully into the running water inside the fume cupboard. The reaction with water is normal and harmless if care is taken. The samples need to be washed with deionized water at least 6 times using the same process as you did earlier while rinsing the acid. However, rinsing should be done inside the fume cupboard until it is sure that the samples are totally neutralised. After the final rinse, empty the remaining deionised water from the centrifuge tubes. The final step of the pollen preparation procedure is to make the slides. Begin by labelling a set of microcentrifuge tubes with the site name, sample number and the depths. Add 1.5 ml of deionised water to the 15 ml conical centrifuge tube. Then, using a glass pipette, agitate the sample to draw the sample into solution and transfer everything into the microcentrifuge tube. Add a further 0.5 ml of deionised water to the 15 ml centrifuge tube to rinse any remaining sample. Add this to the smaller centrifuge tube. Centrifuge the microcentrifuge tubes for 5 minutes and pour off the supernatant. You can tap the tube to try to remove as much of the water as possible. Melt the glycerin jelly in a beaker on a hot plate at low temperature. Label the slide again with all the information about the sample and add a small amount of the melted glycerin jelly to the microcentrifuge tube. The amount of jelly will depend on how diluted you want your slide. It must be at least enough to cover the whole sample. Stir the sample thoroughly with a wooden stick to mix the sample and the jelly. Add more jelly as required. Using the wooden stick, smear the mixture of sample and jelly onto the slide in roughly the form of a capital I. Cover the sample with a cover slip and adjust using a wooden stick until the sample is evenly distributed. Leave the slide for a couple of hours until the glycerin jelly is fully cured. Your slides are now complete and ready for inspection under a microscope.